The Four Classes of Cell Junctions by Chris Rawls, Eddie Mecco, Stephen Guan, and Von Bodecker.
Cells bind together through the different types of cell junction. These junctions create avenues for communication for the cells so that the cells can align and follow the same pattern and express similar cellular behavior. This coordination is important for the body to function correctly and for signals to be passed from one place to the other. There are many different diseases that can cause small disorders in cell junction activity that can have a large-scale effect. Autism is a disease that affects how nerve cells and their synapses connect. Cells can be connected to each other as shown in the figure with a yellow circle, or they can be connected to the extracellular matrix as shown by the red circle. Between these two types of connections and the different functions and signals of cells, this leads us to the four different types of cell junctions. The four different types of cell junctions that we will be examining today include anchoring junctions, occluding junctions, channel-forming junctions, and signal-relaying junctions.
Anchoring junctions are used to hold cells to each other and cells to the extracellular matrix. In connective tissues, there are many more junctions that are holding cells to the extracellular matrix than cells to each other. In epithelial tissues, the opposite is true. There are two types of cell-to-cell junctions, adherence junctions and desmosomes. What distinguishes these two junctions are the cytoskeletal filaments that the neighboring cell is being anchored to. In adherence junctions, this is actin, and desmosomes, the intermediate filaments. Similarly, there are two types of cell-to-matrix junctions, and they are also distinguished by the cytoskeletal filaments that the extracellular matrix is being anchored to. These are actin-linked adhesions, which obviously use actin, and hemidesmosomes use intermediate filaments. There are four main types of transmembrane adhesion proteins: cadherins, integrins, selectin, and immunoglobulin. Cadherin chiefly mediates cell-to-cell adhesion, and integrin chiefly mediates cell-to-matrix adhesion. In this presentation, we'll be discussing mainly cadherins. This is a diagram of cadherin and the protein complex that links it to an actin filament. Classical cadherins have 5 repeated extracellular units, and catenin is the primary protein in the complex that links the cadherins to the actin filament. This is an adherence junction. It is made up of many cadherins linked to the actin cytoskeleton. Individually, these bonds are fairly weak, but packed side by side in parallel, this gives the adherence junction its strength. Adherence junctions typically bind homophilically. This is important in embryonic development when cells of the same tissue type tend to aggregate. The figure on the left is a close-in view of one subunit of the adherence junction. The figure on the right shows the importance of calcium in a cadherin junction. When calcium is present, the cadherin is rigid. When calcium is not present, the cadherin protein is floppy, and proteolytic enzymes tend to degrade it quickly in this state. This is a desmosome. Desmosomes link the intermediate filaments of one cell to another. This gives cell-to-cell junctions substantial tensile strength. Notice that the proteins that link the cadherins in this junction are different than in the adherence junction.
Next, we will be looking at occluding junctions, what their functions are, their types, and examples of how they work in the cell. Occluding junctions, by definition, they seal the gaps between cells and epithelia to make the cell sheet into an impermeable or selectively permeable membrane. In more simple terms, The functions of the occluding junction can be broken down to three primary roles. 1. It holds cells together. 2. It maintains polarity of the cells. And 3. It prevents the passage of molecules and ions. Holding cells together allows for cells to form complex tissues and gain mechanical strength and stability that is essential for the function and survival of the animal. They help maintain the polarity of cells by preventing the lateral diffusion of integral membrane proteins between the apical and lateral basal surfaces, allowing for specialized functions of each surface. For example, this is important in transcellular transport by maintaining endocytosis at the apical surface and exocytosis at the basolateral surface, each surface with its own specialized function. And by preventing the passage of molecules and ions, it helps to force materials to enter the cells either by active or passive diffusion instead of passing through the tissue unchecked. An example of this is the blood-brain barrier, which is essential for preventing harmful bacteria and viruses passing into your brain, which can kill the animal. There are two types of occluding junctions, and they are classified based on their ability to prevent water and solute movement. The first one is type epithelia. It prevents majority of the water and solute movement, while the second type, leaky epithelia, are less complex and have small slit breaks, allowing for some solutes to pass through. And next, we have signal relaying junctions. Signal relaying junctions focus on helping cells pass signals back and forth between each other. The junction allows these signals to flow from one cell to the next across the plasma membrane. This plays a big role in making sure cells behave in a similar fashion. Tying this back to the previous modules, the integrin proteins that are inside the plasma membrane can allow these cell signals to communicate with each other. The first example of this is shown in chemical synapses in the nervous system, where neurotransmitters travel and can cause excitation. The second example is shown in immunological synapses, where T lymphocytes interact with the antigen-presenting cells. This plays into the immune response, also discussed in Module 1 as well. It is also worthy to note that the other three forms of cell junction have important roles in signal transduction, but in different forms. And here is a picture of signal relaying junctions. They involve both anchorage proteins and other proteins that mediate signal transduction.
Channel-forming junctions connect the intracellular matrices of two adjacent cells. They allow for the cells to exchange ions and small water-soluble molecules. In animals, these junctions are called gap junctions, and in plants, they are called plasmodesmata. The building blocks of gap junctions are connexins and enexins. In vertebrates, we predominantly find connexins, which are four-pass transmembrane proteins with a carboxyl and ammonia group in the cytoplasm, and two loops in the extracellular space. 6 connexins form a hemichannel. A hemichannel can be homomeric, meaning that all 6 connexins are of the same type, or heteromeric, meaning the 6 connexin proteins are of different types. When two hemichannels of adjacent cells come together, they form a gap junction, which can be homotypic, meaning the two hemichannels are of the same type, or heterotypic, when both of the hemichannels are formed of different connexins. This diversity in the types of connexins forming gap junctions allows for different permeabilities of the gap junctions. Gap junctions are continuously remodeled in cells. Old gap junctions are discarded from the center of gap junction plaques, and new junctions are added to the periphery through exocytosis. Gap junctions do not remain open at all times. They are pH regulated. In the presence of high calcium ions, gap junctions close. This is particularly important when an adjacent cell dies and its plasma membrane becomes leaky. The influx of extracellular ions in the dead cell would transfer over to an adjacent cell if gap junctions remained open. Through this process, the death of one cell does not lead to the death of neighboring cells. This concludes our discussion on the 4 classes of cell junctions.